Okay, well, good morning, everyone, and welcome to Mohammed Salem's PhD defense. And welcome also to our external examiner from Denmark, that little computer in the corner. And I don't know if our visitors from the US and Egypt have also managed to join us. We'll see. So Mohammed got his undergraduate degree from the German University of Cairo. He did a DSc in pharmacy, and he was part of the first graduating class from that university, and he's going to be the third graduate of that university to graduate from our department. And then he did 1 year at KAUST in the MSc program, but then decided to come and join my group in the fall of 2011. And since that time, Mohammed's been working on a number of projects related to fluorescent protein chromophores and fluorescent proteins. And today he's going to tell us about the culmination of that work as it's been assembled in his thesis, two-photon absorption in fluorescent proteins.
Thanks, Alex. I'll take it from here. So welcome everybody to this 9 a.m. talk. I'll be talking to you about my computation study on two-photon absorption of fluorescent proteins. So on the left we have a schematic diagram for two-photon absorption, and on the right we have a model for a fluorescent protein. And these are the first things I'll try to talk about today. And then I'll talk about the motivation and the thesis outline. I'll follow up with talking, and this would be the bulk of my talk, talking about two main projects of my thesis. And then we'll wrap up with the take-home message and a summary. So to start with fluorescent proteins, I have to project this picture of famous big names up there. So the story started with Professor Shimomura, who discovered fluorescent proteins in the 1960s, and followed by lots of development by Professor Chalti and Professor Roger Chen. And actually, Professor Roger Chen is the postdoc supervisor of Professor Campbell. Professor Campbell is on my committee, so I have some legitimate lineage to fluorescent proteins. Actually, Professor Shimomura is also a pharmacist, so that's great. So fluorescent proteins, they all share this beta-barrel structure. And if you look from top there, this is the top view, you'll find embedded there the chromophore. Chromophore is formed by 3 precursory amino acids, and they have an internal attack here, followed by some dehydration and oxidation, and it matures into this chromophore. And since the chromophore comes from precursory amino acids, then if you change those amino acids, then the resulting chromophore will change in turn. They found that the glycine is pretty conserved in all the fluorescent proteins that they successfully synthesize, but editing, this is edited a lot, the tyrosine, to give full palette of proteins. So we have here, for example, when you change the tyrosine with histidine, you get the blue fluorescent protein. When you change it with tryptophan, you get the cyan. And actually, when you change some of the close-by residues, like here, for example, you get also shifts in the colors. And a very interesting structure that came out later had an extra. So this is like the basic chromophore structure for the green fluorescent protein, but some extra maturation happens here in some of the proteins, and this results in the red-shifted protein. And this is they were very excited when the structure of that protein came out, that they drew it with bacteria on a petri dish in Professor Roger Chen's group. So why are we interested in fluorescent proteins? Because they are fluorescent and they are proteins. So they are proteins, so they are from the native environment of the cell, so they're not foreign to the cell. We don't expect lots of toxicity with using them. And they are fluorescent, so they tell us what's happening inside the cell. If you link it to another protein, for example, then those proteins who were always invisible, then suddenly they become visible. So in the end, we were interested in this range in the light spectrum. But the problem is that we want our proteins to absorb light so that they fluoresce later. And in that range, where all fluorescent proteins absorb, you have lots of competition with other species inside the tissues. So you have, for example, melanin, you have hemoglobin, and from the other side of the spectrum, you have lots of absorption from water. So the first impression here is that absorbing here is really bad. So you have to think about getting closer to this far red or infrared area to have some good absorption without competition. And this is the optical window we want to have our absorption in. Conventional one-photon absorption that everybody is used to, it goes like this. You have a ground state and an electronic state, and then the molecule absorbs a photon that is equivalent to the spacing between the ground electronic state and the excited state. And then it emits a photon, usually of lower energy. So how do we get into this region then? What happens with two-photon absorption, which is the basic idea we're talking about today, is that you get this same spacing, but you cover it with two photons instead of one. So you have the absorption with two photons. So they have, each one of them has half of the energy of this big photon. And so if it has half of the energy, that has double the wavelength. And then if you're having, for example, absorption in this blue region, so like 450 or something, then you suddenly get the absorption in 900. So it's in the region you want. So two-photon absorption appears to be very appealing from this regard. Looking a little bit deeper about what's going on there. So this is a depiction of, here we have a dye, and we have a laser coming from the left and a laser coming from the right. This one coming from the left is at a certain wavelength, and this one is at double that wavelength. And we see that here, one-photon absorption is happening. It's happening all over the path length of the laser beam. Because the absorption is directly proportional to the intensity, and we're having intensity all over this path length. Here we're having photons of double the wavelength of this one. So here, two-photon absorption is taking place. And you see that I'm not sure if you can see this little speck in the middle. So this is where this is the focus of the laser beam. So this is where two-photon absorption is occurring, and then fluorescence is happening. So it's not happening along the way. And this is because of the very nice property of two-photon absorption, is that it's directly proportional to the square of light intensity. And it's just not intensity like in one-photon absorption. And that's why when you have the focus of the huge, intense laser beam, then this is where you get the absorption and you get the emission later. And this is what makes two-photon absorption very natural to image thick tissues. You can get down there with absorption because of your longer wavelengths, and it's actually accompanied by weaker scattering as well. And at the same time, you have a natural focus, so you don't need any focusing techniques. Let's take another deeper look at two-photon absorption. So let's first remind ourselves what intensity means. So intensity is the number of photons passing a certain area in time. So we can give it this unit here. It's photon per centimeter squared for area per time, per second. So in one photon absorption, the units of absorption, like photons per second, they are directly proportional to the intensity, photon per centimeter square second. So this constant of proportionality, which we call the cross-section, has to cancel out this unit here. So it has to be in centimeters squared. And this is very intuitive. This can relate to the area, the effective area of absorption in a certain molecule. And since we're talking about molecules, this number is very small. So we're talking about 10 to the power negative 15 or 10 to the power negative 17 range of area. And this is equivalent to areas of squares of side 3 angstrom to 0.3 angstroms, like what you see in molecules. So let's look at two-photon absorption. We want to apply the same strategy here of trying to derive units for the cross section. So numbers of photons absorbed per second, they are directly proportional to the square of the intensity. So the cross section here, it has to cancel out all those remainders from these units. So we have to have a weird unit for cross section, which is centimeter to the 4 per photon per second. And the orders of magnitude we're looking at here is they are around 10 to the power negative 50 centimeter to the 4 per photon per second. This is less intuitive than thinking about areas here, but this is what we get in the end. And since this is like the basic unit that we have, so we'll be having multiples of this unit. They gave it a nice name, which is GM. This is in honor of Maria Guppert-Meyer, who first predicted the theory that this two-photon absorption can happen. This is in 1931 in her PhD dissertation. So hopefully my PhD dissertation is as good. All right, so now we have some understanding of fluorescent proteins and two-photon absorption. So what is the motivation of this work? So there's a general interest to have fluorescent proteins, or actually probes in general, that have large two-photon absorption cross-sections. So they tend to do this process largely. They have large probability of having this two-photon absorption. One way to do this is to, I'm not claiming that this is the only way to do this, but this is one way to do this, is to use unnatural amino acids to try to edit those proteins, the fluorescent proteins, so you get proteins with some novel properties. And we've got a couple of problems here. The first is that this technique of using unnatural amino acids, and by unnatural we're referring to amino acids that are just synthesized. They are not the 21 amino acids that we're used to. So you sort of trick the bacterial system and or whatever cell you're using, and you express or you include this amino acid in the protein sequence. And then you hope it will be implemented in the chromophore or any other position in the protein, and it will give you novel properties. So this is orders of magnitude of speculation. So it's to start with such a project, and you don't know what you'll get in the end. This is time consuming, and this is effort consuming, and this is expertise consuming. So that's why there were a couple of fluorescent proteins that were made, and then people are tending to stop doing these proteins. There's still a couple of people doing them, but there's not a general interest to use this method, because of its low predictability and its huge experimentally involvement of expertise and time. So the idea is, let's try to guide experimentalists. So we will point out unnatural amino acids that we think, if you use them to replace the tyrosine of the chromophore, then if the chromophore matures, then hopefully this will be a chromophore of very, very large two-photon absorption cross-section that you can engineer and work on later. The complexity of the problem is that there is the two-photon absorption of that chromophore is affected by many things. First, its intrinsic ability to, its intrinsic cross-section to do this two-photon absorption. And second is that we need to remember that the chromophore is not free in solution. It's surrounded by a protein shell, which directs its conformer. So this change in conformer also affects the two-photon absorption cross-section of the chromophore. And third, because of the protein shell as well, this exerts an electric field. It has an electric field that affects the two-photon absorption of the chromophore as well. And actually, it's found that in two-photon absorption, specifically, this third layer of complexity is very important, because it really affects the two-photon absorption of the chromophore, the electric field and the electrostatic interactions with the shell of the protein. Nevertheless, in my research, I just used those two layers of complexity, and not yet did I touch upon the third level. And now let me take you to my thesis. It starts with some introduction of using two-photon absorption in biological molecules, and this is based on a book chapter that Melis and I wrote under the supervision of Alex, of course. It was published in the Handbook of Computational Chemistry. And then chapter 2 is a computational comparison between some of the techniques that are used to predict the two-photon absorption and to study them in fluorescent protein chromophores. I will not be talking about this. This was published in JCTC in 2014. I'll be mainly talking about chapter 3 and 4. And chapter 3 is just the screening of green fluorescent protein-based chromophores and studying their predicted two-photon absorption. And chapter 4 is very similar to this, with some more analysis on red fluorescent protein-based chromophores. And this was resubmitted 2 days ago. Hopefully it will get through. And this is, of course, wrapped up by a conclusion. So I'll be focusing on chapters 3 and 4 in the remainder of my talk. So let's start with the screening of GFP-like chromophores. First, the way we prepare the chromophores, this is if you remember, this is what I started the talk with. So this is the GFP chromophore. I have a model for it here, and we'll return to it. So this is not the scale. So this part is the part that comes from the tyrosine. This is the tyrosine side chain. And this part here is what comes from the threonine or serine, and this part comes from glycine. And what we're doing is that we're cutting all the branching to the protein. Actually, I'm missing a hydrogen here. Yeah, we're putting. Okay, just pretend there's. Okay, I found one. So, and we're putting hydrogen caps. So we're removing the connections with the protein, and we're putting hydrogen caps. And then we're changing this part, so that is expected to come from the tyrosine. We're changing it with a set of unnatural amino acid branches. So these are side chains of the unnatural amino acids. And you can see that we chose them to be a little bit similar to the tyrosine, with all of them having those aromatic regions. We got this pool from the work of Liu and Schultz in 2010. And the reason why I got this pool is that all of these were actually used by experimentalists to make proteins. Some of them for fluorescent proteins and some of them for other proteins, but we're not just speculating. Those were actually used unnatural amino acids. So to look at this one here, this side chain, I have it here. And I'll be using it in some of my discussion. And this one was actually used in a fluorescent protein that is called the gold fluorescent protein. So the way you should think of this is that you should attach this piece to this piece in there. And there is other unnatural amino acids that were used. So those three here, and all of these three, amongst with this, were synthesized by Peter Schultz in 2002. This is where I was in high school. And he showed that these three are fluorescent, while this one was not fluorescent. But in the paper, they rationalized this, that they need to engineer the cavity a little bit more so that they can accommodate it so it matures properly. But in the end, this is the set I'm having. So we built those chromophores, and then we went on optimizing them in gas phase using this DFT functional and 631 plus GDP basis set. And we went on computing excited state. We heavily relied on TDDFT. We used B3DIP functional in the beginning, and later we used CAM B3DIP as well. And we used 631 plus GDP basis set. And we used the model for water, which is a PCM model, a continuum model for water. There is no explicit water, but it's just a dielectric constant around our chromophore models. And we also used gas phase. So we used both. And we computed 2-photon absorption cross sections, 1-photon absorption energies, and 1-photon absorption cross section or oscillator strengths. And here are the results. And you see that there are some chromophores that are not doing quite well, and there are chromophores that are actually having very large 2-photon absorption cross section. And to have some sort of comparison here, if you do the green fluorescent protein chromophore, if you do this one here, and you compute the 2-photon absorption with the same method, you'll get around there. So this is what we did. It's around 5 GM. Sorry. So if you compare this to the nitro-based chromophore, let me borrow this nitro from here. So what we did is that we remove this oxygen, we put a nitrogen. This is a little bit more difficult to do experimentally. And so this is the chromophore. This is the nitro-based chromophore. And you find that this chromophore is having a very, very large 2-photon absorption cross-section. This is like 7 times the 2-photon absorption cross-section that you expect from the green fluorescent protein chromophore. The energy, concerning the energies, I didn't put all the energies here, but this is, actually the energies were sort of decreasing as you go to the right, which is a good thing, because when the energies decrease, it means that the wavelength is increasing, so you're getting into the redder region. And always think that, remember that these are to the double. So this is, if you convert it to wavelengths, this is around 420 nanometers. So the double of this is around 840 nanometers, so you're in a very good range. And this one here, this is for the gold fluorescent protein that I showed you with this part instead of the tyrosine side chain. And this one, I'm very happy with this because the energy for this one is, the experimental energy is 2.66 electron volts, so we're very, very close to the experimental energies. Let me show you the CAM B3DIP results. And you see that in general, we're having a similar trend. Of course, there is some anomalies, but in general, we're having some sort of a similar trend where we get the small ones, the small TPAs in the right position, and the large TPAs in the chromophores with large 2-photon absorption in their on the right. Now let me stick to B-predict for now. This chromophore model, chromophore number 22, the nitro-based chromophore, is the winner with us in the GFP screening. And this is what we continue to work on for this study. So what we did afterwards is that we said, okay, if we take this one and we put it inside an EGFP shell, or a green fluorescent protein shell, what would happen to the protein? So we tried this with molecular dynamics, and we took an EGFP crystal, an enhanced green fluorescent protein crystal, and we mutated it the way I did, by replacing the oxygen with the nitro. And we prepared this for use in a molecular dynamics software package, which is AMBER. We used a pretty standard scheme of solvating the structure in a water box, and then running this with minimization, heating, density equilibration, and then a trajectory of 99 nanoseconds, which is okay trajectory. So these are the main results that we got. Here we have a plot of the root mean square fluctuations in each residue. And you see the red belongs to the nitro modified protein, and the control is the EGFP original crystal. And you see that the red is actually following the black quite well. So there is no exaggerated fluctuations anywhere in the protein, except here in regions where regions that are not close to the chromophore. So those are outer loops that might be less interesting in determining the stability of the region surrounding the chromophore. And actually here, this is an overlay of the red refers to the nitro again, and the black refers to the green fluorescent protein crystal structure, where you have, you see that the structures are nearly overlapping. This is an average structure over the main course of the trajectory. Second point was thinking what happens to this chromophore when it's inside the protein pocket. So how does it change its conformation? We looked at three factors here. So we looked at three angles over around 50,000 snapshots over the 99 nanosecond trajectories. And the angles are, if you look here, so this is, I always drew it this way. So the angles we're looking at are first the methine bridge, so this angle, I'm changing here. And we're looking at two rotating angles. So we're looking at rotating this ring, which I call this tilt, and we're looking at rotating this ring, and I call this twist. So you see here the methine bridge, this one, it appears to be quite rigid. So it's having a peak, a surge here at around 140 degrees, this angle, I'm not sure if this is 140, this is not to scale. And then when you study the twist on the right, you see that it's a little bit more flexible, but most of the conformers we sampled, they are having angles between 160 and 200. And here we have twisting from 30 to minus 30. This is what covers most of the conformers that we had inside the protein pocket. So what we did is that we took our optimized structure and we fixed it at a methine bridge at this angle of 140, and then we systematically rotated the conformer and we sampled two-photon absorption. So we will fix, for example, this twist angle here at the planar position, and then we change these by 5-degree increments, and then we compute the two-photon absorption, and then we change this a bit, and we fix it, and then we rotate this again. And then we compute the two-photon absorption at each of these values. So we get a 2D, we get a 3D actually a plot here where we have the z-axis is the two-photon absorption, and here we have the tilt and the twist, and we don't have the methine bridge because it's fixed at 140 degrees. And you see that when you change this is here the tilt angle of where it is in the planar position, and this is where you get the maximum two-photon absorption. Once you go away from the planar position, then the two-photon absorption decreases. But this is not the case for the twist angle. You see that the twist angle we prefer it to be twisted. So as it goes away from the planar position, the two-photon absorption increases. So now we want to look a little bit. We want to have a little bit deeper look at why this is going on. So what is causing the nitrochromophore to have this large two-photon absorption? What is causing the others to have smaller two-photon absorption? And we'll relate this first to the one-photon absorption, and we see that this doesn't look very nice. We cannot relate these to these. So we see that the chromophores that are having good or relatively okay one-photon absorption, they don't have good two-photon absorption. And this is one lesson that is well learned before, is that we cannot predict two-photon absorption from one-photon absorption. So what's going on? Let's look at the two-photon absorption process a little bit deeper, but within a two-level model. And by two-level model, we mean that we're looking at the ground state and the first electronic state, and we're not involving any other state. Actually, to have a better picture and to have the right picture of two-photon absorption, we should involve more states in our analysis. But let's keep it at this level. So what are the factors that affect first one-photon absorption? It's only one thing. It's the transition dipole moment. So when the molecule is being excited from the ground state to the excited state, the first excited state here, the thing, if this transition dipole moment is large, then there is more probability of having one-photon absorption. In two-photon absorption, we're also concerned with other factors. So we're concerned with the difference between the permanent dipole moments of the excited and the ground state. The ground state has a dipole moment, we all know this. But actually, when the charge redistributes in an excited state, it also has another dipole moment. The difference between these is a factor that affects two-photon absorption. If you have large difference between the permanent dipole moment of the excited state and the permanent dipole moment of the ground state, then you will get large two-photon absorption. So to sum up, factors affecting the two-photon absorption are the transition dipole moment, just like one-photon absorption, and the difference between excited and ground states, permanent dipole moments. And everything is squared. So it varies with the square of the transition dipole moment and the square of this difference between permanent excited state dipole moment and ground state dipole moment. I want to remind you that we're doing this in two-level model, but actually to have the better picture, you should do n-level models. But this is very good to grasp the basic understanding of what's going on. So let's look within a two-level model. So those are the plots I showed you before. This is the two-photon absorption cross-sections for all chromophores, and this is the one-photon absorption cross-section or oscillator strength for the chromophores. And here are the computed values for the difference between permanent dipole moments in green and transition dipole moment squared in relatively black. So I'm just cheating because I want to give you the impression that this one is similar to this and this one is similar to this, which is okay, but it's easier to see this way. And you see, of course, that the transition dipole moment is perfectly matching the oscillator strength, because this is basically computed from this. And you see that the two-photon absorption cross-section is following the difference between the permanent dipole moments of the excited and the ground state. And we need to remember that these are vectors. Dipole moments are vectors. So what I'm referring to here is actually the x-axis of the dipole moment components. And you see that the analysis is going okay with the x-axis, with this going along the main axis in the molecule here, along this methane bridge. You see for model 22, which is the winner, the nitro model, that it has a large difference between permanent dipole moments and a large oscillator strength as well, a large transition dipole moment, and that's why, relatively large, and that's why you have this largest two-photon absorption. So this wraps up the discussion on the reason why we have large two-photon absorption for the nitro. But now let's look at the behavior that we saw before for the change in two-photon absorption with conformational changes. So we see again that this is the curve that I showed you before, and this is the curve for the difference between dipole moment squared, and it's perfectly matching this curve, which means that this is the main driving force, the change in the dipole moment differences. This is the main driving force for this trend in two-photon absorption cross-sections with conformational changes. And we see here, this is the oscillator strength, which is very different from two-photon absorption trend. So now we're done with the GFP-like scan, the GFP-like chromophore scan. And we want to repeat the scan for RFP-like chromophores, which is the next product. And I want to remind you here, with what I started with, is that the green fluorescent protein is made by these 3 precursory amino acids. And in other proteins, we have an extension here. We have an extra maturation step, and we get this structure. So actually, what we did is that we took this structure, and we also broke the connections to the protein. And we got a structure that looks like this. A main difference that we're seeing here, so this is my GFP-based chromophore. I lost an oxygen. So the main difference we're seeing here is I'm taking off those hydrogen caps, and I'm putting the extra acylamine moiety. I hope it fits. Here, and it's rotating. And then I capped this one here with methyl group and not a hydrogen. So this is the basic structure that we're having. And of course, we changed this part here that comes from tyrosine. And when we change this, we get all the chromophores that we're studying. So let's put it in the red area. And we followed a very similar scheme, that we have an optimization followed by the excitation, by computation of excited states. And here this is where we reported all the B3lib versus the CAM B3lib values. We did it in PCM and gas phase, and we computed the 2-photon absorption cross sections and the 1-photon absorption properties. And this is what we got. And here I'm putting the red fluorescent protein derived data with B3lib and with CAM B3lib. And again, we're having a sort of similar trend. But we're seeing here that the winners are a little bit different. So this was the one with the nitro in the GFP-like chromophores. And we see that the ones, the number 20 and number 21 are having the most, the strongest 2-photon absorption cross-section. And to remind you, model 20 is actually the one formed by, I'm losing my structures. It's now we have to remove this part that we got from the, doesn't want to go. The part that we got from the tyrosine, okay? And then we have to put this part that is similar to the gold fluorescent protein. So the gold fluorescent protein chromophore will end up till here. But now we have the extended acylamine moiety that we added which is characteristic for the red fluorescent protein chromophores. So this is based on a red fluorescent protein template, and it's based on the gold fluorescent protein. So this is model number 20, which has the largest 2-photon absorption here. Let's look at the gas, no, this is not the gas phase. Let's compare first the red fluorescent protein ones to the green fluorescent protein ones. And we see that the red fluorescent protein ones are much, much larger than the green fluorescent protein ones in terms of 2-photon absorption cross-section. So we focused more on number 20 and model 21, which is having a 2-membered ring as well in the position of the tyrosine side chain. So the conclusion we're having here is that it doesn't really matter if you use gas phase or if you use a model with large dielectric constant, you'll get the same trends, and we're actually focusing on trends in here. And now let's look within a 2-level model again on what is causing this large increase in 2-photon absorption cross-section for those red fluorescent protein derived chromophores. And I want to remind you that the 2-photon absorption cross-section is directly proportional to the transition dipole moment and to the difference between permanent dipole moments of the excited and ground states. And I want to remind you that these are vectors. And there is a nicer model to look at these, which includes the, so now we have them in their vector form. And to use these in the previous project, I used only the x-axis. But now if I want to use the magnitude, so I'll be using the magnitude of the transition dipole moment and the magnitude of the difference between permanent dipole moments, I have to include the angle between those vectors. And you see from here, cosine theta, is when the cosine of the angle is 1, it means that they are more aligned. And when it's closer to 0, it means that they are not aligned. We don't care about their directions, but we care about their alignment. If they are more aligned, then they have large 2-photon absorption cross sections. So the first thing I started with is to try to calculate all these terms and to calculate the 2-photon absorption with these terms. So I show you here in purple the calculated two-photon absorption using the two-level model. And I show you the red trend here is the one that we computed with B3LIP using the previous method that I showed you in the previous slides. And you see that it's working okay. Of course, there are some differences, and these differences is because we're not including we're only included two levels in the model. And I told you that to get the very proper trend, very proper absolute values, you have to include more than just two levels. But in the end, we're getting the correct trend. So the two-level model sort of works, and this is what we need. So here, this is again the same plot that I showed you in the previous slide, and I want to remove the quadratic response values, and I'll keep the two-level model values. And again, these are the one-photon absorption. These are the transition dipole moments that give large one-photon absorption, and these are and this is the difference between two-photon absorption and one-photon absorption is that it depends on this value. And you see that there are molecules here that have expected large one-photon absorption, but they don't have two-photon absorption. But now when we look at the alignment, so those ones here are the models that have aligned dipole vectors, and here they are perfectly not aligned. And you see that for such model, for model 10, for example, it has a large dipole difference, and at the same time it doesn't have large two-photon absorption cross-section for two reasons: for having very low transition dipole, and for having vectors that are not aligned at all, which blows off the expression of two-photon absorption. So we looked, let's go through this quickly. So we looked at the rotation of at some of the conformational changes that might happen to the chromophore inside the protein. And the first thing we looked at is the rotation of this extended moiety. So we have here an acyl imine moiety. So we looked at the rotation of this one with respect to the whole molecule. And we saw that this one has an impressive effect on the two-photon absorption. We see that it's having an oscillatory trend that goes from maximum when it's planar to minimum when it's perpendicular. Let's focus on one of these, which is model 21, the one I have up there. And let's look at the difference that we're having here. We're having a change from 70 to 40 GM. So this is a huge change. We're looking at in this and other models, we're looking at 40 to 50% change in two-photon absorption due to this change in acylamine dihedral. And let's take a deeper look again at what is causing this change. So we calculated this with the two-level model, and we're having the correct change, so we're good. And then we calculated the parameters that are affecting this change. And again, it follows the trend in the difference between permanent dipole moments and not the transition dipole moment. We studied the same tilting and twisting angles that I showed you before, but now with model number 21, and we got those figures here. What is interesting to look at is that the difference is not big. So whatever the trend is, but the difference is we're looking at from 70 to 64 or 65 GM. These are 5 GM units. Compared to 40 GM units, it looks like the acylamine moiety is the one that is playing the biggest role in here. So I'm close to the end. So the take-home message here, there's some general take-home message, which is always when you're looking at computational data, try to know the strength of your method. So I'm not trying to lie and say that I believe all the absolute values that I computed here, but I can claim that the trends are okay. And this is the strength of computing two-photon absorption for my chromophore models. The second thing is that we have to remember the approximations. Here I'm completely ignoring the electric field of the protein. So we're just computing the two-photon absorption within a chromophore, the intrinsic to that chromophore, and we're looking at how it would change with conformations, but we have no idea what would happen when we put it inside the protein. But the bright side is we've seen that engineering proteins through engineering proteins, you can affect two-photon absorption and you can amplify it a lot. But they were working on natural proteins, red fluorescent proteins and green fluorescent protein like structures. And these, when we computed their two-photon absorption, it's very minimal. So for the GFP, it's around 4 or 5 GM, and for the red fluorescent protein, it's around 7 GM. And we're now looking at molecules that we claim they have 7-fold of increase in two-photon absorption. So if you take these and you work on them, you invest time on them, then probably you'll have better two-photon absorption probes. The sensitivity of the method, our method, it doesn't depend that much on the choice of function as long as you're just concerned with the trends and the choice of the medium, like we've seen with using gas phase versus water model. And then we can use the two-level model to get some physical insight of what's going on with why we're having those trends of two-photon absorption, what is causing the amplification, what is causing the loss of two-photon absorption. And, and we looked at the two-level model, and it also explains the change of two-photon absorption with conformational changes. So today I talked to you about fluorescent proteins, and I tried to entertain you with two-photon absorption. I told you that we're trying to predict chromophore structures with large two-photon absorption. I told you about the, I talked to you about the screening of green fluorescent protein-like and red fluorescent protein-like chromophores. And for the future work, there is some need to do a red fluorescent protein molecular dynamic study, similar to the one I did for the GFP, just to sample those acylamine dihydrals properly, and to include protein effects, which I've been ignoring. And this can be done with having a bigger chunk of the chromophore, not only the part I was using, but the chromophore and some surrounding residues, or even the whole protein, but in a mixed hybrid QM-MM fashion. And we need to validate our method a little bit more, like to have better trust and better faith in our absolute values, and we're actually working on this now. And finally, the message is for the experimentalists. They should take it from there. So they have some good structures that I'm claiming they have good properties if you synthesize them. So let me end up with saying that I had lots of fun, and I'll give you one example of this fun. This is an email I sent to Alex once in 2013, and I was like happy with my rhyme. And then this is the response I got from Alex. So he was using a rhyme as well. So we're having this type of environment in our group, and this is really fun. But most important is that you have to notice that Alex draws a nose here when he makes happy face, and I don't do this. Thanks for all those fun years. And thanks for the members of our groups. All these are doctors by far, except for Melis, hopefully soon. And thanks for the undergraduates in our group, Katun, the hardworking Japanese that is always there and with that. And Wenyu, is he around? Yeah, Mr. Wenyu. Thanks for your work on the GFP project, which I failed to convert it into a paper. But Wenyu is a really hardworking, and actually for an undergrad that was pretty impressive. You leave it to him, and you know that he would do it right. And Isaac worked with me in the last project that I showed you, and this is turned into a paper and is being published in Focus. And thanks for Ahmed Abdel Fattah. He's now in the US doing a postdoc. He helped me a lot with some of the reality stuff concerning fluorescent proteins. And Ahmed Ayub here, he's helped me with molecular dynamics. Thanks a lot for everybody.
Thanks, Mohammed. And now, I guess there are time for questions from the non-committee members, if anyone has questions.
I know it's a hard question, but what would sort of be the theoretical maximum of a two-photon absorbance? Like the structure you found there, is that anywhere near the best possible chromophore structure you could stuff inside of a fluorescent protein, or is there still a huge range of improvement if you were to make?
All right, so the best approach to answering this question is first to remind you how I did these. So I did them with the pool that is already there, right? So if I give myself the liberty to just try to, to improvise and to come up with a better structure, I'm not sure how I will get there. There are lots of studies on how to maximize two-photon absorption for isolated molecules. But when I look at the structures, they have some symmetries and stuff like that that is hard to put in two-photon in fluorescent proteins. I'm not sure how it would work. So the short answer to the question is I don't know. But this is the best I got with the structures with screened.
You used to show the cross-section results from B3 lib and CAM B3 lib, and you used to build on B3 lib, on CAM B3 lib. Is there a reason for that?
So there's a strong debate. I think this will come up in the defense. But the main reason why I trust B3 lib is that it computes better energies, and I don't trust the absolute cross sections of the of whether you do it with B3 lib and CAM B3 lib that much. And since we're getting the same trend, so I just went with the one that computes better energies.
I'm just curious, on your on the green fluorescent protein, when you tag the phenyl ring with a nitro group, did you attempt to maintain the angle of the ring steady, but this rotate the nitro group?
I didn't rotate the nitro. So when I did this, you mean when we had the so back to my models. So when we had the benzene here, and we had the nitro there. So this part was kept planar. So what I played with is rotating this part as a whole.
So my question is because typically in whether it's solid or liquids, the aromatic ring can do a rotation, and it's usually quite rapid in liquid phase or in solid. And I'm wondering if the response that you're seeing when you go from your angle, your twist angle from 100 to, or it was 120 even, 160 to 200, if it's due to the fact that the nitro group is being affected by it, or if it's actually the aromatic ring.
So first to comment on the first part of your question is that the yes, in solution phase this guy rotates a lot. So you have lots of rotation here, and that's why those chromophores are not fluorescent in solution phase. But now we're having them rigid in the protein environment. And what I did is just I sampled what's in there, because they are rigid, but of course it's a dynamics thing, and they are having some space, and I actually freed the space a little bit so to make it more flexible. So what I did is that I sampled this flexibility that is going on here, but I didn't really think about what's coming from the nitrogen. I just looked at these. I didn't even look at the pictures, so I just sampled the angles from the trajectories. Hope this answers your question.
Okay, we should probably let the committee get their hands on Mohammed. So let's thank Mohammed one more time. And